Blood vessels are going to have three tunics, whether it is an artery or a vein. Capillaries will be a little bit different, and I'll talk about those in a minute. But you're going to have to be able to tell me what these three tunics are and what's included in each tunic. So we're going to start with the inner tunic that lines the lumen. Remember, the lumen is the hollow portion of a tube. This is what the blood is flowing through. We'll start with the tunica intima. It's also known as the tunica interna. The term intima comes from the fact that this is the layer that is in intimate contact with the blood. It's going to consist of a simple squamous endothelium, so that's simple squamous epithelial tissue there, a subendothelial layer, as well as an elastic membrane. The tunica media will be the middle layer, and this is going to be typically your thickest layer, especially if we're talking about arteries. It's going to consist of smooth muscles and elastic fibers. These are the smooth muscles that are involved in vasoconstriction and vasodilation, and they will be innervated by the sympathetic nervous system. If you increase sympathetic input to the smooth muscles, vasoconstriction will occur. The smooth muscles will contract and the lumen will get smaller, or the diameter of the tube will get smaller. If sympathetic input decreases, vasodilation will occur. And you're also going to have an external elastic membrane in the tunica media. Moving outward, the outer layer is called the tunica externa. It's going to be connective tissue, so you're going to have some collagen fibers there. Recall that collagen fibers have great tensile strength, so this means that they provide some reinforcement for the vessels to prevent them from blowing out. It's like having a stocking around them. When you have large blood vessels, the tunica externa is going to have its own blood supply, and those blood vessels are called the vasa vasorum. So you can see them in the tunica externa. They're the little blue and red dots there. The yellow dots represent nerves. Vasa vasorum actually means vessels to vessels. Let's look at the capillary down at the bottom. Notice that the capillary is only going to consist of an endothelium and a basement membrane. Recall that all epithelial tissue rests on a basement membrane, and this is what connects it to the underlying loose connective tissue. So endothelial tissue always, or epithelial tissue always has a connective tissue associated with it. In this photograph, you can see the artery on the left and the vein on the right. What I want you to notice is the thickness of the walls, the size of the lumens, and the shape of the vessels. So if you look at the artery, you'll notice it has a thicker wall. It has a thicker tunica media. The lumen is smaller and it's nice and round, whereas the vein will be partially collapsed. Smaller wall. So there's the artery, and there is the vein. If you look at this, let me go back, what's highlighted. See if you can figure out, what the kind of vessel that is. That is an artery. Look at the lumen and look at the thickness of the wall. This vessel here, I'm going to go back so that you can actually see it. That is a vein. So the vessel I have highlighted is a vein. Look at the wall. You'll notice how the wall is much thinner. You'll have to be able to identify on a histology slide whether you have an artery or a vein. And in this case, this is going to be an artery. The three tunics are fairly visible here. This is going to be the tunica intima, the endothelium. It's actually the lighter pink area there. It looks like a little line of light pink. Notice how it's scalloped. So Recall that I said smooth muscles are slightly contracted so that you can contract them more for vasoconstriction or relax them for vasodilation. And so that little scalloped edge next to the lumen is very characteristic of arteries. This is the tunica media. Notice how thick it is. So those are all smooth muscles. And then you have the tunica externa. Hopefully you can identify this type of tissue from AP1. This is adipose tissue. In this photograph of a histology slide, try to figure out what vessel this is. Is it an artery or a vein? It's an artery. And this is your vein. So notice how much larger the lumen is, the fact that the vein is partially collapsed and the wall is much thinner. And you can see the tunica media in the vein is much thinner than the tunica media in the artery. There are several different types of arteries. Elastic or conducting arteries are the arteries that are attached to the heart. They're going to have the thickest wall, but they're very elastic. And so this accommodates blood when it's being ejected from the heart during systole. These vessels actually expand due to the elasticity or the elastic fibers. And then during diastole, the blood continues through the vessel and the vessel recoils. So you have a very healthy vessel. It's going to stretch and recoil in relationship to systole and diastole. And that's what you're actually feeling when you feel somebody's pulse. So these vessels include the aorta artery and the pulmonary trunk artery. Remember, the aorta artery drains the left ventricle and the pulmonary trunk artery drains the right ventricle. So elastic arteries or conducting arteries offer very little resistance because they have so much elasticity. They can stretch to accommodate the blood as it's being ejected from the heart and then recoil when the blood continues downstream. The second type of arteries are your muscular arteries, and these are the arteries that have names to them that you're going to be identifying. So these are going to be your medium and your small arteries, the ones that are feeding your organs. And then the smallest of your arteries are called arterioles, and these are the ones that are going to feed your capillary beds. These are your resistance vessels. In other words, these are the vessels that are undergoing the vasoconstriction and vasodilation, and they're going to influence blood flow. If you have vasoconstriction of the terminal arterioles feeding the capillary beds, then that's going to decrease blood flow into the capillary beds and redirect the blood into the main arteries. With the capillaries, as mentioned before, they're going to consist of a simple squamous epithelium layer that's called the endothelium, as well as usually a basement membrane. With veins, you have the larger medium-sized veins, and then you have the venules, and these are the smallest of your veins. And these are the ones that drain the capillaries or receive blood from the capillaries. Recall, arteries carry blood away from the heart, and veins always carry blood toward the heart. That's what defines whether you have an artery or a vein. It's not whether you have high oxygen or low oxygen in the blood. The aorta is going to be richly oxygenated since it's receiving blood from the left ventricle. This blood has returned back from the lungs. The pulmonary trunk artery, on the other hand, will be poorly oxygenated. This blood is coming from the right ventricle and is being sent to the lungs to pick up oxygen and get rid of carbon dioxide. So remember the rule. In the systemic circuit, arteries will be red and veins will be blue. So that means the arteries are going to be rich in oxygen and the veins will be low in oxygen. And this is just the color on the model. In the pulmonary circuit, arteries will be blue and veins will be red. That's because in the pulmonary circuit, the pulmonary trunk artery receiving blood from the right ventricle is taking the blood to the lungs, so the blood hasn't gone to the lungs yet to pick up oxygen. So arteries just carry blood away from the heart, and veins carry blood toward the heart. Let's look at some differences between arteries and veins. Veins, as mentioned before, are going to have a thinner tunica media, so their walls look thinner. Their lumens will be larger, and they'll be partially collapsed. In addition, veins will have valves, and that's because the pressure in the veins is very low. Recall from lecture that the pressure in the vena cava veins is almost 0 millimeters of mercury, so extremely low. You have to have valves to prevent blood from flowing backwards, and these valves are very similar in structure to the semilunar valves. If you look at the photograph on the right, you'll notice the probe is in one of those valves. So they open like swinging doors, just like the semilunar valves. And then when the blood goes through the valves, as the pressure drops and the blood starts flowing backwards, it fills the little cups of the valves and keeps the valves closed. So these valves make sure that the blood keeps returning back to the heart. So they prevent backflow. Let's look at capillaries. Capillaries will have tight junctions between the endothelial cells. So if you look at the upper illustration that I've drawn, I have two cells in green. The orange little balls, those are proteins that fuse together between two adjacent cells, and that creates a tight junction. And notice how that forms a barrier. So substances cannot go between the cells since you have these proteins there as barriers. So that's my tight junction. The number of tight junctions that you have varies depending upon the capillary type. Intercellular clefts are just the spaces in between the two endothelial cells. So those are in green. The cleft is at the end of the purple arrow in that illustration. Substances are going to move across the capillary wall by diffusion, which is passive, bulk flow, which is passive, and penocytosis, which is active transport. Associated with capillaries are contractile stem cells that are called pericytes, and in the illustration here on the right, these are the green cells there. These are interesting cells. We're just learning more about them, but they seem to be important in maintaining the blood-brain barrier. They also, and they control permeability there, so they help control that. It seems that their relaxation will allow vasodilation of the capillaries. So they do have contractile fibers, and that seems to have an influence on blood flow within the capillary itself, and this is unrelated to the arterioles. In addition, they can generate new blood vessels and stabilize the capillary wall, so they have several different functions. There are 3 different types of capillaries based on the anatomy. The illustration on the right of the 3 capillaries that's from your book, so I would direct you to that. It's got some good information in there. But let's look at the 3. The one at the top is a continuous capillary, and this is the type that you're going to find in the blood-brain barrier, also the thymus blood barrier. These are the least permeable of the 3 types of capillaries. You're going to have a lot of tight junctions. The intercellular clefts will be very small, and the basement membrane is going to be complete. Fenestrated capillaries have fenestrations, which are pores, and you can see one at the end of the green arrow. So this is the illustration in the middle. these are going to be more permeable. You'll have fewer tight junctions, and also the intercellular clefts will be slightly larger. You can see these in the kidneys, making up the filtration membrane. And then sinusoids. These are going to be the most permeable of your capillaries. We see this in the liver. If you look at the illustration, notice that the basement membrane, which is at the tip of the green arrow, is incomplete. You also have very large fenestrations, those pores, and you have large intercellular clefts. So these are very permeable. This is comparing the two capillaries. The one on the left is actually a continuous capillary, so you can see between the two cells, the intercellular clefts are very, very small. And then on the right is fenestrated, and at the tip of the green pointer, you're going to see a fenestration or a pore. How do you study? Begin by looking at the flow charts in your textbook. Learn those flow charts so that you know how the blood flows. After you've done that, you can then begin to draw your own flow charts. Keep drawing the flow charts over and over again until you can draw the flow charts without looking at your notes. After you've done that, then look at the models. You should be able to identify blood vessels on any model. It doesn't make any difference what model I show you, or on an illustration. Learning the flow charts will help you do that. Learn the blood vessels in body sections. You could learn the blood vessels in the trunk, the upper appendages, the lower appendages, and the brain. So take this in small amounts. Be sure that you know the vessels in one part of the body before you move to the other. You'll learn the arteries first, and then the following week, you're going to learn your veins. Blood vessels may be named, and often are, according to where they are located. You're going to draw on the terminology that you learned in AP1. This will become very useful. Many arteries and veins run parallel to each other and have the same name. Time to get started.